All right, so the talk I'm going to give you today is, well, it's entitled, hold on, I'm trying to hide my video panel here. Hide my floating meeting controls. There we go. So it's entitled, Can hippocampal neurogenesis be a marker of affective valence in poultry? It's a very open-ended question. What I'm going to tell you is some of the evidence from rodents that indeed hippocampal neurogenesis might be an indicator of valence, then tell you about our work in chickens, which is mostly on the negative end of valence, and some of our more recent surprising results that hopefully will be of interest to you as well. And we're still kind of making sense of that. But before I start, before I show you that, let me set the picture. So, as you probably all know, animal welfare is a very many-faceted concept. And most animal welfare assessment, definitely in practice on farms, is actually based to date still mostly on resource-based indicators rather than animal-based indicators. So, making sure the animals have access to food, have access to certain resources, have access to water that they need, have enough space, have enough enrichment, all these kinds of things. So these are not indicators based on what the animals look like, but what the animals have, what we give to the animals. Alternatively, people do use some animal-based indicators of welfare. Health ones are very obvious ones. So if your animals are in a good, healthy physical condition, it's very likely that they're in much better welfare than if they're in poor physical condition. That everybody would agree on that. But ideally, what we would also have are some animal-based indicators of affective states, so of their general emotional state. And in animal welfare, we often look at affective states in this conceptualization here, this multivariate or multi-axis conceptualization of affective states. So in this particular case, it's two axes. One axis is arousal, so going from very activated to very deactivated. The other axis is valence, going from unpleasant to pleasant. And for animal welfare, we really care about the valence axis because more and more there's an interest in the concept of positive animal welfare in the animal welfare field, which means it's not good enough that the animals aren't undergoing negative conditions. In an ideal world, the animals would undergo mostly positive states or would experience mostly positive states. So there's a recent definition that positive welfare is a predominant experience of predominantly positive states. We don't assume that they will always be positive, but more positive than negative. And also an inclusion of having more resilience, for example, to stress. So those are aspects of positive welfare that people care about. Now, arousal is a dimension that's fairly easily measured. In fact, the HPA or SHPA axis that Greg just talked about and the sympathetic nervous system response are all parts of that arousal response. But those increases in activity and arousals can be positive or can be negative. So an excitement of a calf going out to play in a meadow will be high arousal, but positive valence. But an animal being, I don't know, herded somewhere or grabbed by a vet for inspection or something along those lines will also be highly aroused, but possibly on the negative side. So arousal is not a very useful indicator when it comes to animal welfare. Indicators of valence would be very useful, but they're not easy to come by. So if you want a good marker of valence, they should at least have these two qualities. So in the first instance, they should co-vary with the valence of the experience, positive and negative. So a good indicator should go up, let's call it, when the valence is positive and down when the valence is negative. Could be the other way around, of course. Could be down when the valence is positive and up when the valence is negative. As long as they go in opposite directions for positive and negative experiences, that would make it a good marker of that valence. And I say here experience because it shouldn't just be an indicator of the conditions that the animal is exposed to. It should be an indicator of the response of the animal to those conditions. So it should be, is that condition positive to that particular animal or negative to that particular animal? And we can all imagine that there might be conditions where different animals respond in different ways, positive for some, negative for others. That could be based on their history, their developmental history, their personality type, their genetics, all kinds of reasons why a particular experience could be positive for one animal or negative for another. Now, the hard part of that is, of course, is to know, if we want an indicator, what would be, if you want a good indicator of positive or negative experience, somewhere you need a gold standard, right, of what is positive and what is negative. That one's still the hardest one. I think in animal welfare, the most common definition of an indicator of something that gives a positive experience is something the animal is willing to work for. Something that gives a negative experience is something the animal is willing to work to avoid, something along those lines. But that requires a lot of training, a lot of testing of individual animals. It is not something you could easily scale up to larger numbers or to different conditions. So what I'm going to tell you about is what I think is a potential biomarker of valence, or at least what we thought to now as might be a potential biomarker of valence that is anatomical, neuroanatomical, rather than requiring lots of training and testing of animals in a behavioral way. So it's adult hippocampal neurogenesis. So I'll break this down for you. I'm going to tell you about a bit of a literature in rats and mice, first of all, because that's where the inspiration came from, and that's where there's a lot of literature on this, mostly from the literature on preclinical models of depression, essentially, anxiety and depression. And so, this adult hippocampal neurogenesis is usually reduced in these models of anxiety and depression, and that's where the whole idea started. So let me first tell you what adult hippocampal neurogenesis is. So what you see here on the left is a diagram of a mouse or a rat brain, and in pink in the middle there, so it's the brain is transparent, and there inside those hemispheres sits the hippocampus on both sides. Notice that unlike in humans, the hippocampus in rats and mice does not look like a seahorse. It looks like a cashew nut or a banana, if you want to think of it that way. Importantly, there's two ends to that. Let's call it a cashew. There's two ends to that cashew. The septal pole connected to the septum, that's why we call it that, which the literature is getting clearer and clearer about, is mostly involved in memory processing and navigation, so more cognitive function. Whereas the other end, the temporal pole, seems to be more involved in the regulation of the stress response and of emotions. So it's one neuroanatomical structure, but it seems to have multiple functions, and they are neuroanatomically separated from each other along that long axis of the cashew. Now, if we cut through this cashew, cross-section through it, we get something like this. Obviously, this is not the cashew, but it gives you the concept. So if we cut through the hippocampus, what we see is this dense layer of neuronal cell bodies that we call cornu ammonis. Anna talked about this. And then this arrowhead on the one end of it, where the CA3 area is, that arrowhead, which is a dentate gyrus. And so this is really this, what we see here as a line is really a cross-section of a sheet that is rolled up like this, and another sheet that is folded up around the tip of that sheet. Yeah, so think of this in 3D going in and out of your screen. Now, the adult hippocampal neurogenesis is making of new neurons in the adult hippocampus, and in mice and rats, adult is quite young, often can be a few months old, up to a year or two, and the numbers do go down with age for sure. But in the adult rodent hippocampus, in the dentate gyrus specifically, in this bottom cell layer, which is called the subgranular zone, this red line here, is where new neurons are born. And so if we zoom in on this bit here, We here have that subgranular zone. Here we have the rest of the cell bodies of the dentate gyrus. And so we will have in there a number of different cell types that start with the stem cells, which will divide and bud off daughter cells. Those daughter cells will then divide several times. So these are the amplifying cells, and those amplifying cells then eventually start differentiating into neurons, starting as immature neurons, eventually becoming mature granule cells that are indistinguishable from the other granule cells in the dentate gyrus. Now, it's this process here that we're interested in. And so what I'll show you in the next slide is two things. One, I'm going to show you that the number of these newly generated cells indeed is sensitive to chronic stress. And secondly, I'll show you a number of different ways in which they are quantified, because that's actually also important for the rest of the story. So what we see here is just an image from a study by Yun et al. From 2010. It was nice because it showed three different ways of measuring adult hippocampal neurogenesis. I hope you all recognize here that arrowhead of the dentate gyrus in all these pictures. In the left column are the control non-stressed mice. In the right column are the stressed mice. And what you should see immediately is in each pair that there is fewer black dots or black marks. I'll tell you what this is in a minute, in the stressed column than in the unstressed column. Ki67 is a marker, an endogenous marker. It's a protein that's involved in mitosis. And so it is expressed in dividing cells. So there's more of these dividing cells, which are either the stem cells or the proliferating cells in that subgranular zone. You see it's just on that one layer of cells. And there's fewer of those in the stressed animals. BRDU is not an endogenous protein. BRDU is a chemical that you inject into the animal, and it gets incorporated into the DNA during the S phase of mitosis, but then it stays in there. So it doesn't just mark dividing cells while they're dividing, like Ki67, it marks the dividing cells, and they are marked for the rest of their life as having divided during the time when the BRDU was in the system. So when BRDU had been injected into the animals. Let me see if I can make the pointer better. There we go. Then finally, double cortin is an endogenous protein, and it's made by immature neurons. And so it's not just in the nucleus like the other two, but it's expressed throughout the whole dendritic arbor of these neurons, because it is part of the cytoskeleton and of the growing out of the new dendrites in the immature neurons. And that's why we see so much more staining here than in the other two. But again, there's less staining in the stressed animals than in the non-stressed animals. So that's what we're looking at. We, in my lab, work with double cortin, but I'll show you some results first that use some of these different markers. Right, where are we? So the first thing I want to show you is, well, this one just showed you that chronic stress reduces adult hippocampal neurogenesis. So the negative end of the valence axis. And notice that I'm talking about chronic stress here. So we're definitely talking about long-term affective states here, not acute affective states. But when we talk about long-term affective states, there's loads and loads of evidence that chronic stress reduces these cells in the rodent hippocampus. But there's also some evidence that positive conditions increase these numbers of cells. So I'm going to show you a little bit of that. So the first thing we want to see here in this paper by Lehmann et al. From 2013, it's a very complicated figure. So I want you to focus just on the comparison between that black line and that black bar. Sorry, that black bar and that black bar. So these are home cage animals. These are social defeat animals. So this is the chronic stress treatment. This is the control. In this particular study, they also took the adrenal glands out. That's the other color. But what you should see here, that's the rostral, the cognitive bit of the hippocampus. This is the emotional bit of the hippocampus. And in the emotional bit of the hippocampus, chronic stress reduces the number of BRDU labeled neurons compared to home cage. Yeah, so that's the main event I want you to see here. So chronic stress reduces hippocampal neurogenesis. In the same paper, they also gave the animals enrichment. And of course, there was also another group that had sort of home cage or social defeat, and then no enrichment, home cage just afterwards. And so what you see, what the delta means is higher than the ones that have no enrichment afterwards. And so enrichment increases neurogenesis in the caudal dentate gyrus and somewhat now also in the rostral dentate gyrus. So the first finding for this paper, and I apologize for the really complicated figures here, is that chronic stress reduces neurogenesis, enrichment increases neurogenesis. Yeah. What they showed in the paper here as well, this is what all the other bars are, is that if you remove the adrenal cortex and therefore prevent corticosterone from being naturally raised at all, so they basically clamped corticosterone at low levels, then none of those effects happen. So you need corticosterone to reduce the number of new neurons in response to chronic stress, but you also need corticosterone to increase neurogenesis in response to enrichment. And so that very nicely follows from the previous talk, saying be careful how you interpret the result, increased corticosterone, because it may well be a negative thing, but it may well be a positive thing as well, or indeed just a physiological adjustment, like was said before. Okay, so, but this is the first piece of evidence, and there's lots of evidence now that enrichment increases neurogenesis in the hippocampus. What to me was the most convincing evidence, however, is this one. Here we have one of those rare, almost gold standards. I'm going to say almost because I'm not going to say completely gold standards, but almost gold standards in positive experiences. Rats like to be tickled. And in fact, you can tell whether a rat likes to be tickled, because if it likes to be tickled, during the tickling, it'll make these 50 kilohertz calls, which are thought of as the rat laughter. If it doesn't like the tickling, it makes the 22 kilohertz calls, which are negatively valenced. And so we can tell here, not only that most rats like tickling, but also from an individual basis, whether that animal is experiencing the tickling as a positive or a negative experience. And in this study here by Yamamura et al., they showed that tickled rats have higher number of, in this case, oh, I forgot what they stand for. Yes, BRDU positive cells. This means neurons, this means non-neurons, but either way, both kinds go up with tickling. And in the Ver et al. Study, they show that only the rats that laugh, quote unquote, so that make the 50 kilohertz calls, get the increase in neurogenesis, but not the ones that get tickled but don't like it. So that is a very nice link between a gold standard, their vocalizations, direct experience of the individual, and our indicator of positive or negative experiences. And so in rodents, I am fairly, well, at least in rats, presumably in mice, tickling of mice is not a thing. They definitely don't like it the way rats do. So in mice and rats, we can tell that adult hippocampal neurogenesis goes down with negative experiences, up with recovery from negative experiences, including an enrichment, and up with positive experiences, like in the rats, like with tickling. Okay. So we do seem to have an indicator here that follows positive or negative experiences. What's less clear is a time course of all these things, because even though there's loads and loads of studies on chronic stress and adult hippocampal neurogenesis, there have been very few, or I can't think of any, that have systematically followed how long it takes before this indicator starts following or before it can reverse. So there's a lot of missing information there if you really want to start using it as an indicator of how much positive or negative experiences this particular animal has had in its lifetime. Okay. Now, all of this is rats and mice so far, so I really need to move on to poultry, because that's what the Poultry Extension Cooperative is all about. And as you've already heard before, I don't need to convince you probably that hippocampus isn't just a rodent thing. It's not even just a pig thing or a human thing. There are homologous structures in the brain of every vertebrate species that are fairly well established as homologs of the hippocampus, including in birds. So, and more recently, it's also become clear that not only is there a homologous structure, it also looks like that functional separation along the long axis of the hippocampus from more cognitive on one end to more emotional on the other end seems to also be conserved, at least between birds and mammals. There's not been any work done on this in any other vertebrates. And so when we're looking in birds, we're looking at the anterior, which I'm going to call the rostral hippocampus as the more cognitive end, and the posterior, what I'm going to call the caudal hippocampus as the more emotional end of the hippocampus. That's important because all the results I'm going to show you from our lab always split the hippocampus by these two sections, the back end and the front end. Okay, so first question we asked in our lab, we needed to validate that hippocampal neurogenesis in chickens was indeed an indicator of chronic stress. Did it respond to chronic stress the way it does in rodents? So what we did an 8-week treatment of unpredictable chronic mild stress on Highline brown hens. And the unpredictable chronic mild stress is indicated here in this picture here. So on any given day, they got a different stressor. They never knew what was coming from day to day. And so the stressors we had, we isolated them in a box for a few hours, or we put a fan in their room, which gave them lots of wind, which they don't seem to like. We took the food away for a while. We sprayed the bedding and left it overnight, which started smelling quite badly. We increased or decreased the temperature in the room, and we manipulated the photoperiod. In fact, our stressed group had an unpredictable photoperiod for the whole of the 8 weeks. So the average photoperiod per day was always the same, but in unpredictable blocks of light and dark for the stressed birds. And the control birds didn't have any of these things and had a very predictable normal photoperiod the whole time as well. What we counted were these kinds of cells. So these are cells stained for the protein double cortin. So DCX stands for double cortin. Double cortin in rats and mice, as I told you before, is a protein involved in this, well, it is definitely a protein involved in the cytoskeleton and remodeling the cytoskeleton. It is definitely expressed in immature neurons. Whether those immature neurons are always newly generated neurons is a bit of a debate in the literature. They definitely are in rats and mice. Whether they are in chickens might depend on the age of the chickens, and there's still a little bit of debate there. So let's call it immature neurons for now. So what did we find about these immature neurons in the hippocampus in chickens? Well, here's the rostral, so the cognitive end of the hippocampus. Here's the caudal or the emotional end of the hippocampus. These are Z-scored cell densities, which is why we get negative values. But what we see is a large difference between our control group and our unpredictable chronic mild stress group in the caudal hippocampus, but no significant difference in the rostral hippocampus. So much lower density of these immature neurons in the birds that we experimentally induced stress in, chronic stress in the caudal hippocampus. So that's validation that in birds as well, in chickens as well, 8 weeks of unpredictable chronic mild stress does indeed reduce the density of these immature neurons, and especially in the caudal hippocampus, which is the bit that we expect to be most sensitive to stress and emotions. So it looks like we can use these levels, these densities, as a marker of chronic stress, negative valence, at least severe chronic stress like this unpredictable chronic mild stress. So the next thing we look at then is whether a quote unquote naturally occurring, or at least industrially occurring stressor, it can also be detected by this method. And so we looked at keelbone fractures. Now you're all poultry people, so you know about keelbone fractures. So a few x-rays here of keel bones with more fractures going in that direction. I am not the x-ray specialist. We did this in collaboration with Mike Toscano, whose lab I'm actually in right now in Switzerland, doing a collaborative study right now as well. And so what Mike did here, he had a study ongoing in which the birds were living in their multi-tier aviaries, like here. In this particular case, I believe the brown birds were the focal birds, and they put them among white birds to more easily find them. And then they X-rayed them 11 times throughout their whole productive life to see when they developed these keelbone fractures. And we came in at the very end and collected brains from a subset of birds with severe keelbone fractures and a subset of birds with mild keelbone fractures. We could not find enough birds without keelbone fractures by the end of the laying period to have a no keelbone fracture group. What did we find? So first of all, I haven't told you before that we actually count two separate morphologies of cells. One are called multipolar, and that's what you've seen before. So cells with multiple processes coming out. The bipolar morphology is a bit more so long-looking cells. So it has just two processes 180 degrees apart. And in the bird literature, these are believed to be migrating neurons. So young neurons moving towards where they're going to establish themselves, whereas multipolar neurons are supposed to be neurons that are in the place where they're establishing themselves and now outgrowing out their dendrites to make connections. So just to show you, to explain why there's two bits to this graph. But in both of them, you see the rostral hippocampus and caudal hippocampus, rostral and caudal. Again, minimal keelbone fractures in white, severe keelbone fractures in black. We have the Z scores again, which is why we get positive and negative values. But you see, especially in the multipolars, we see that there's a significant difference between the minimal and severe keelbone fracture birds, with the severe keelbone fracture birds having lower cell densities of double cortin. And that difference is now, in this particular case, it's significant in the rostral and the caudal end, but it's larger in the caudal end. So that's why we get this interaction between subregion and keelbone fracture group for the multipolar cells. For the bipolar cells, the overall effect of keelbone fracture is significant. It's just not different in the two bits of the hippocampus. So the bipolar cells still respond to the keelbone fractures, just not differentially in the two ends of the hippocampus. So what we have here is clearly a reduction in these double cortin cells by the, or at least correlated with, to be strictly correct, because of course we just selected the birds with keelbone fractures. We didn't induce keelbone fractures in these birds. What was really nice is we also, because we had the 11 X-rays, we could look back and see how long ago a bird acquired its first keelbone fractures. And when we did that, you could see that the birds that had acquired their keelbone fractures the longest ago, so the first X-ray, second X-ray, third X-ray, fourth X-ray, etc., in their life, had fewer double cortin positive cells than the birds that only acquired their first fracture later in life, in the caudal hippocampus, but not in the rostral hippocampus. So in the caudal hippocampus, there's a clear correlation between the time since the first fracture and the density. So the longer they've lived with these keelbone fractures, the fewer of these double cortin cells they have, at least multipolar ones. So in the caudal hippocampus. So again, this suggests that the longer lasting the stress, the fewer of these net cells we have. So it is an indicator not just of a negative experience, but potentially there's an indicator there of the dose of that negative experience, or at least the duration of that negative experience. So keelbone fractures suggest that indeed this known stressful and painful condition can be picked up by looking at the density of double cortin positive cells, and that the longer they've lived with it, the fewer of these cells do they have. So we have an indicator here of potentially the severity and the duration of a chronic stressor, but of course it is still a negative valence state. In fact, I'll own up to the fact that we still haven't done positive experiences in chickens. I would really like to do this, but we haven't got funding for that at the moment. So it would be interesting to see if we can get this number of double cortin positive cells up with some kind of positive intervention. Because we haven't yet, we have started using it to look at the negative side. And so I'm going to show you two more studies in which we've now asked the question, are certain conditions more negative than other conditions based on the double cortin density as our measure? And the first one of those is housing systems. As you probably all know, there's a lot of movement happening at the moment. Europe is about a decade or a bit more, a few decades maybe, ahead of North America in this move from cages and traditional battery cages to begin with. I think those are definitely, well, in Europe, they've completely been phased out, or at least haven't been legal since 2012, to a completely cage-free system. And so what you see here on the left are free-range birds that inside have a multi-tier system. On the right, you have the colony or enriched cage systems that are also still in use in the UK, and I'm assuming in North America as well. So we were curious because, of course, as an egg consumer, what we hear all the time is free range is so much higher welfare than cages. We should definitely all have free range eggs. So we were curious to see if we could actually see that in the hippocampus. Is there a difference between free range birds and colony caged birds in the double cortin densities in their hippocampus? So that was our question. Now, because within every system, you get healthy birds and you get scrawny birds that are not so healthy, we decided to sample from the two housing systems to sample high quality, good physical quality birds and poor physical quality birds to kind of capture that variation within each system, rather than randomly grab some birds and you might end up just with some good quality birds in one system and poor quality birds from the other system. So we made sure we picked some top and bottom birds, 12 of each in each system. And so the good physical condition birds had good feather cover, high body weight, red comb color, compared to the poor condition birds, which were very scrawny, so you could feel their keel bones much more prominently, their combs are much lighter, their feathers condition is much poorer. What did we find? So first of all, what you're looking at here again is rostral and caudal. This time rostral and caudal is Z-scored together, which is why the rostral numbers are all negative and the caudal numbers are all positive. The last few times what you've seen is they were Z-scored separately. But what you see is within each of them, the gray ones are the poor condition birds, the pink ones are the good condition birds, and then the darker ones are the free range birds, and the lighter ones are the caged birds. And we found no effect of housing system whatsoever in this. So free range birds versus caged birds have similar densities of double cortin in rostral and caudal hippocampus. We're talking multipolar cells here, but there was no different pattern in the bipolar cells at all. And what we did find is that within each system, poor birds have a lower density, poor physical quality birds have lower density of double cortin cells. So what that suggests to us is that from the bird's perspective, it's a negative valence to be a poor physical condition bird, probably not too surprising compared to a good physical condition bird. But once you've lived, and we took them from commercial settings, and these were birds that had lived in those systems for their whole lives, fairly old birds. So from the bird's perspective, there was no difference in the double cortin density, whether they lived in enriched cages, colony cages, or whether they lived in free-range systems. So no difference between housing systems from what we can tell, at least where we were looking in the hippocampus in the measure that we take. So it's if there are differences between the systems, they're smaller than the differences between poor and good quality birds. They're also much smaller than the differences between birds with keel bone fractures and birds without keel bone fractures. So the welfare implications might be, if you really want to compare housing systems, maybe you should just go in and count poor and good quality birds, because that might give you a much better overview of which system might be overall better welfare for the animals than the system itself. And then finally, we looked at a potential other stressor, which is footpad dermatitis. So we thought, well, if keel bone fractures are stressful, what about footpad dermatitis. And so we see the three pictures here of the three scored levels of footpad dermatitis. So none in this one, some necrosis in this bird, and quite a lot of necrosis and swelling in this bird here. And so that's how they were scored 0, 1, and 2. Again, we worked in collaboration with a colleague in Belgium in this particular case, who had birds that were going to have to be culled. Many of them had footpad dermatitis. So he said, why don't we take some brains from the ones with footpad dermatitis and those without, and see if it really is a chronic stressor for us. Now, unbeknownst to us when we sampled, but told to us after we had already processed the brains, some of the birds that we took way back in their history, because we took birds at the end of lay, but way back in their history, some of them had been reared with dark brooders and some had not. Now, I don't know how familiar you are with dark brooders, so I'm going to quickly introduce dark brooders to you. Dark brooders are essentially a system in which during rear, especially the first weeks of life, the birds are given access to these dark brooders. Here's one of them. And they can basically go into those flaps and come back out. So the birds have full agency to go into a dark and warm place and come back out from that dark and warm place. It's supposed to be somewhat mimicking what a mother hen can provide. So cover, darkness, warmth. But of course, even if it's not what a mother hen can provide, it gives the chicks agency to be somewhere warm and quiet if they want to sleep, and can go run around outside of that area in the light and the cooler area if they want to be active, and they don't interfere with each other that way. So whereas in a general rearing barn, if one animal's trying to nap and the one next to it runs over it, there's nothing they can do about that. Dark brooders, if nothing else, separates the space in such a way the animals have agency about where they go, and that in itself might make a big difference. There's lots of literature on positive impacts of dark brooders, especially in the young birds. And so we decided that we better take that into account in our analysis, because it was not a source of variation we had originally planned on. So what happened then? So what you're seeing here is the densities, again, Z-scored, of these double cortin cells: bipolar cell type, multipolar cell type, caudal hippocampus, rostral hippocampus, control group, footpad dermatitis group, but also split by whether they were reared conventionally or with dark brooders. Okay. And I want you to look at this panel here, because it's where you see the effect best. But what we're looking at is an interaction between footpad dermatitis and rearing overall. So that's independent of cell type and part of the hippocampus. Is just because these are all on the same axis, it's much harder to see in these other ones because the numbers are just smaller. So look up here. So what you should see there is, remember, conventionally reared are on the left, dark brooder reared on the right. And hopefully what you're noticing here is that in the conventionally reared birds, we see the expected effects. The footpad dermatitis birds have a lower density of double cortin cells than the control birds. But in the dark brooder reared birds, we see the opposite effect. The footpad dermatitis birds have an increase in double cortin density. So that's why the interaction between footpad dermatitis and rearing is significant. Okay, so that was unexpected. But it made us think about, even if this is not a real increase, it is definitely not a decrease like here. So even if we assume that this is flat and this is a decrease, that suggests early rearing with dark brooder might be somehow giving the birds resilience to the stressor of the footpad dermatitis, or making the birds more able to cope with that footpad dermatitis. So this was an unexpected result. So that, Matt here, whose PhD, this was the first study of his PhD, then decided to kind of make this the topic of his PhD and look at dark brooders, but in a proper way. And so his next experiment was to actually manipulate a chronic stress and dark brooders so we knew what was going on. Okay, so summary here was that we have this strange effect of dark brooders. So let's have a look at whether we can do that experimentally. So he had a 2x2 design. We had generally the same chronic stressors as we had in our first experiment. And now we had birds that were either reared or not reared with dark brooders or not. We got the birds from a commercial farm in the north of England who actually have commercial dark brooder rearing sheds. So we were able to take birds that had been reared in the dark brooder sheds and other birds that had been reared in a nearby non-dark brooder shed from the same, exactly the same age, because they'd all been placed at the same time and from exactly the same genotype, so the same type of birds. So we took 12 of each and then exposed 12 of each, sorry, we took 24 of each and exposed 12 of each to chronic stress and 12 of each not. To make a long story short, we don't have double cortin densities here. We have gene expression because we had a -80 freeze or disaster that got rid of all the brains, but luckily we had also dissected the hippocampus from the other hemisphere and we could do qPCR for double cortin in those. Now, long story short, if you look in the caudal hippocampus, we see a similar effect to before. In the conventionally reared birds, the UCMS, the unpredictable chronic mild stress, reduced the density of, the gene expression of double cortin, whereas in the dark brooder reared animals, it increased the gene expression of double cortin. So where does that leave us? If we take a strict valence interpretation of double cortin, it suggests that the dark brooder reared birds consider unpredictable chronic mild stress and maybe footpad dermatitis, although that was a flatter pattern, as a positive experience, which we find slightly difficult to believe. And so that means that maybe that interpretation isn't right for chickens. So how do we interpret it? So now we get to the very speculative and, we're still thinking about this stage. So what about this? What if, like many other features, the density of double cortin positive cells responds in a non-monotonic way to stress? So increases for manageable amounts of stress and then decreases for stress that what people might call allostatic overload, when there's more than the system can handle. So in that case, we would say that our conventionally reared birds might have started here and the chronic stress moved them there, decreasing density, and maybe the dark brooder reared birds started here and the same amount of stress increased their neurogenesis. That's one interpretation. Another possibility of thinking about it is that maybe dark brooders change the response shape. Maybe it stretches us out. So if all the chicks start here and then we give them stress, it moves the conventionally reared birds into the zone where they can't cope anymore. But because the dark brooders have a wider zone of coping, they're increasing their neurogenesis because they're still coping with the stress. So that's another possibility of how we interpret our findings. So is it a marker of valence? Probably not reliably if those last two results can be believed, at least in chickens. So what is it a marker of? Well, now we're really speculating. We're speculating based on mice again. And so here in this study in mice, and I will just quickly tell you what this study says rather than talk you through this through the slides, because I realize we're running out of time. But essentially what the study in mice showed was that if you experimentally, and in mice you can do this genetically, manipulate the number of new neurons in the hippocampus, you can show that, especially, this is in the stress end of the hippocampus, you can show that if you experimentally reduce the number of those new neurons, the animals are more sensitive to chronic stress and will develop anxiety with levels of chronic stress that normally wouldn't trigger anxiety. And if you experimentally increase the number of new neurons, you can make them resilient to chronic stress that would normally trigger an anxiety response in the mice. So in the mice anyway, new neurons in the emotional part of the hippocampus seem to be causally involved in stress resilience. So maybe something like that is going on in the chickens as well. And maybe what we're seeing with the increase in neurogenesis in the dark brooder birds is an indication of a resilience response. And the animal, as I said, coping with that resilience, that stress whereas the conventionally reared animals may very well not be able to cope, be beyond their resilience, and therefore we're seeing that reduction in double cortin positive cells. So maybe it's not a direct indicator of valence, but maybe it's an indicator of how resilient and how well the animals cope. And that is still an aspect of positive animal welfare and still a useful thing to know how animals cope with negative experiences. What it doesn't give us, though, is a good indicator of positive experiences. And so that we will still have to check how that works for this system. So wait and see, I guess. I hope that was interesting to you all, and I want to thank all my collaborators. I'm not going to go through them all, but these are all people that I've worked with over the last 10, 15 years on these projects. Thank you very much.